Okay, real quick, for me, exciting disclosure of conflicts of interest. So I'm on sabbatical right now, as of yesterday. So don't I look well rested? I'll be working as a research scientist at Meta Reality Labs in New York, but today I'm wearing my Emory and Georgia Tech hats, not my Meta hat. Okay, so let me tell you about using AI to power sort of what we hope to be the next generation of brain-computer interfaces. Okay, so what are the applications that we're talking about here? There are millions of people with limited movement from conditions like spinal cord injury, stroke, neurodegenerative diseases like amyotrophic lateral sclerosis, ALS, cerebral palsy, Parkinson's, and often these conditions cause people to be reliant on caregivers for some of their most basic needs. So the goal of brain-computer interfaces are to restore movement to this population by reading out signals directly from the brain through implanted electrodes and translating these signals, often using machine learning and artificial intelligence, to do things that are useful for the person. So let's imagine that the person wants to imagine writing. You might decode their handwriting. Maybe reaching and grasping, you could have them control something like a robotic arm, or speaking, you could decode that type of activity and translate that into speech. And it's a really, I think, exciting time for brain-computer interfaces. In the academic realm, there have been a lot of recent successes, so things like being able to write in sensory information for people who've lost that ability, restoring control of voluntary limb control of paralyzed limbs through functional electrical stimulation, things like point and click typing and computer control, or rapid typing through decoding handwriting. And I think this exciting wave in the last couple of years here has been decoding intended speech directly from the brain. So quite a few exciting pieces of work in that space. And it's also a super exciting time in industry for brain-computer interfaces. And I haven't updated this slide in a year, and I could probably double it with like the number of companies that have come about lately, including some really exciting ones like Motif. I hear they're doing some great stuff. So I think for us in academia, I mean, what a privilege to be able to have kind of this industry option for our trainees, as opposed to studying something for a long time and then going off to do something completely different. Now there's a way to translate a lot of these things that we've been developing to actually make a difference in people's lives. Sorry, this pointer isn't quite there. And of these, maybe the most one that gets a lot of media attention. Elon Musk's Neuralink group is already in clinical trials. They just implanted their second participant. So I mean, I think earnestly, I think it's a very exciting time, and Neuralink is doing some really exciting work. So I just wanted to show you a little bit of our work on speech decoding. Most of my talk is going to be on motor control, but this is a bit of a tease of some stuff that will be coming out in the next year, because last year, with my partner Nick Aoyong, a functional neurosurgeon at Emory, we started our clinical trial for brain-computer interfaces at Emory. We're working with participant T16, who unfortunately has paralysis due to brainstem stroke. So of strokes, brainstem stroke is particularly severe because it interferes with a lot of movement. So for her, she has very limited movement of her hands. She can move her mouth to speak, but it takes a lot of effort to push any air through, so it's very effortful for her to speak. So one of the things that we've been working on is we implanted electrodes. Nick implanted electrodes into motor and speech regions of her precentral gyrus, and we're decoding that activity, in this case for a speech application. So I'll just show you. What you'll see here is what we're asking her to try to say. You'll see her actually, She'll move her mouth to try to say it, but she's not trying to push air through because it takes so much effort for her. And then you'll see kind of in real time, the system, just based on brain activity, decode what it is that she's attempting to say.
How many divisions does he have?
So I'm not going to go too much into the architecture here, but it's pretty standard, kind of machine learning for sequential data. So we're using recurrent neural networks and bidirectional configuration, predict phoneme probabilities, and send that
long-term consequences.
Send that into a large language model and ask it, to clean up things after kind of that direct decoding. But as you can see, it actually does quite well in not quite real time, but and you can see it. Well, actually, it's doing so well that it didn't really need to update.
We were talking about that just the other day.
But for somebody that has such limited communication options, I mean, for somebody with brainstem stroke, they might be confined to their bed or their chair for most of the day and have very limited ability to interact with people. These types of devices could
now that I've seen him,
could be life-changing. Okay, so how does this work? Intracortical brain-computer interfaces give access to neuron spiking activity. So this is an example of the types of electrode arrays we use in the clinical trial. These are Utah arrays. You can see a thumb in there in the background to give you a sense of how big one of these is. So this has about 100 electrodes on it, and it records voltages from neurons as they communicate with each other. You see these little blips. These are action potentials. So these are the activity of individual neurons as they communicate with each other. So you can think of the signal that we get out as basically a binary code of just 0 most of the time, and then 1 every time we detect an action potential. And of course, we need to translate that into what it is the person wants to do. So I'll just show you like a little clip of what activity looks like from one of these arrays. This is actually from a monkey array, and you can kind of see these large voltage deflections here. That corresponds to, those are action potentials as neurons in motor cortex are kind of talking to each other as the monkey's moving its hand. So the neuron's activity, it actually represents thought, right? It represents the intention to move. And so our challenge and the applications of AI here are, how do we decode this activity into the movement intention? And not just that, but because we need to be responsive to that activity, we need to do it in real time. We need to decode on the fly. And I'll also say it's a particularly exciting time in neuroscience because our ability to interface with the brain has been changing really substantially over the last few years. So what I just showed you was the activity of about 100 electrodes. And this is from Ian Stevenson and Conrad Kording, where they track the number of neurons that people report in the literature being able to simultaneously record. And it's a little out of date, but you can see that the Y scale here is a log scale, and this is a linear trend on a log scale. So this is exponential growth. So recording capacity is doubling about every 6 years, probably even faster than that now. And so this is a huge opportunity. The data are just getting more and more complex, and they're really information rich. And so AI provides new opportunities to understand these data. So I'll very quickly go over some of our work, a new scientific understanding of these data, which we call neural population dynamics, which provides a new way to interpret movement. I'll show you how combining AI with dynamics can lead to high-performance decoding of movement intention. And then I'll also show you how we can attack a different problem, which is the robustness of brain-computer interfaces, also using AI. Okay, so let's imagine that we're going to, I told you that we can record from hundreds or thousands of neurons. Let's simplify the problem for just a minute and imagine that we're only recording from 3 neurons. So neuron 1, neuron 2, neuron 3. And these little ticks represent the action potentials from those neurons. And let's say, this is a movement, and you asked a monkey or a person to make many movements, what you get is some sort of smooth firing rate. So kind of like the average across many movements looks kind of like a smooth curve. And so this is for 3 neurons. You could imagine we could make a state space plot here where each axis represents the firing rate of an individual neuron, neuron 1, neuron 2, neuron 3, so that any point here corresponds to a point in our state space. Okay? So empirically, what we're finding as we start to study these data is that activity doesn't just kind of span these spaces uniformly, as if these neurons were acting independently, but rather there's clear structure to the neural activity. So for example, it may occupy lower dimensional manifolds. So lower dimensional than 3 in this case would be a simple plane. And you might see that, go over a long time, but activity stays confined to that plane. And another really important feature that we notice is that activity doesn't just kind of randomly zip around these manifolds in time, but there seem to be structured rules to how activity evolves. And we call these rules dynamics. And the basic idea here, I think this is maybe the only equation I have in my entire talk, so you can burn a little bit of brain power on it. The basic idea is, so if the state of the system is S, then we're saying the change in state is some function of the current state. Meaning kind of if you know where you are in space, you can predict what's going to happen next. And the rules that govern that are kind of depicted graphically by this vector field or by some function, a nonlinear function that tells you how activity evolves over time. Okay, so an example of where this has been shown in the past was some great work by Mark Churchland and John Cunningham when they were in my former mentor, Krishna Shenoy's lab, where they trained a monkey to make a large number of reaching movements. So the monkey is kind of making reaches in a 2D workspace. These are all the different types of reaches that the monkey would make. And then they recorded neural activity from about 200 neurons in motor cortex. And it looks something like this, like I said. So that kind of binary code, imagine 200 dimensional. This is not bright enough. But imagine, 200 dimensional by time. And then every time the monkey reaches, you get something that looks like that, but different depending on what the monkey's doing. So what they showed is if you take that activity and do dimensionality reduction to find this underlying manifold that I was describing, so that kind of state space approach I showed you earlier, so monkeys are making a variety of reaches. Over here are the trajectories in neural state space that they found from the neural activity. So corresponding to each reach here is some trajectory in the neural state space. And the dots represent where the activity was just before the monkey starts to move. So the monkey knows where it's going to go, and so it's kind of planning to get there. And so that separation is represented here, so that's why the dots are separated. And then when the monkey decides to actually move, you see this trajectory. And the really amazing thing here is that all of these trajectories, despite the wide variety of reaches that the monkey is making, they all seem to follow a very simple set of rules, which is kind of, if you know the starting position, you just kind of rotate. The counterclockwise-ness is arbitrary. You could just flip this plot and it would be clockwise. But the point is really the consistency across the wide variety of behaviors that the monkey is doing that tells us that there's something predictable there. And if we can learn these rules, we can better understand neural activity. Okay, so again, that kind of vector field are the dynamics. And this is highly simplified. I said there are 200 neurons here, so if I show you a 2D plot representing 200 neurons, you should know that I'm sweeping some things under the rug, but it's still actually a good depiction of the neural activity. Okay, so how can we use this to improve brain-computer interfaces? So as I said, changes in neurons' firing rates are coordinated. And we see that population activity kind of exhibits these dynamics. What I didn't tell you was in order to pull out these trajectories in this paper, they had to basically ask the monkey to do a movement many times and average the activity over all repeats of the same movement. So, when you just look at activity, it looks like these kind of binary blips, right? But if you have the monkey do the movement many times, and you smooth it, and you average it, you get something that's also smooth and has all this structure. But that doesn't really help us for brain-computer interfaces, because we need them to be responsive on a moment-by-moment basis, right? This is a closed-loop control problem. We need to be responsive in real time. So we can't ask you to try to grab the cup 20 times before we move the robot to grab it for you. We need to be able to respond to your activity in real time. So working with David Cicillo and some other collaborators, we took as a starting point this idea that activity has underlying dynamics. So we're not questioning that. We're actually just saying, let's assume that the dynamics are there. How can we pull it out without averaging? How can we pull it out on a single trial, a single movement basis? And so we want to take that kind of spiking activity that I was talking about and translate that into these underlying trajectories. And so the way we're doing this is we're assuming that for every movement, it corresponds to an initial state in our state space. And then across all movements, there's one set of rules, those dynamics, that govern how activity evolves over time. So, we reasoned that if we could learn those rules, then for any given movement, we just need to know where to put it in that state space in the beginning, and then we can just kind of integrate over this flow field to pull out these underlying trajectories. And so how are we doing that? We're doing this with a classic machine learning approach called an autoencoder, which takes high-dimensional activity, compresses it down to a latent representation, and then tries to reconstruct the original high-dimensional activity. This is often used in images, high-dimensional activity like 1,000 by 1,000 pixel image. That's a million-dimensional signal, right? So the difference here is like images are static, but this has time in it. So we don't use what you might use in common autoencoders like feed-forward networks or something like that. Instead, we're using recurrent neural networks, which are really good at picking out temporal patterns. So we have an encoder recurrent network that takes the spiking data, maps it down to a vector. And we have a second recurrent network called a generator, which only gets that vector and has to reproduce the high-dimensional time-varying activity. And so in order to do that, it needs to learn that vector field. It needs to approximate those dynamics. And the cost function here, we're not trying to reconstruct the spikes exactly, but we're assuming there's a smooth underlying firing rate and just assume a Poisson likelihood. And so this is all randomly initialized. We train these networks through back propagation, pretty standard, classic deep learning at this point. And so what do we get? So I showed you what Mark Churchland and John Cunningham got when they averaged over many repeats of the same movement. We do the same thing with our spiking activity. But now each of these lines represents neural activity on a single movement, right? This is about a 250 millisecond window, 2,100 trials. So the monkey's just reaching in a completely unsupervised way. The system has gone through and tried to find structure in that data, and this is what it comes up with. So we didn't tell it that there were these rotational dynamics, but rather it found those same dynamics in order to best describe the data, but now it's able to do that on a moment-by-moment basis and on a millisecond timescale. So how does that help us for BCIs? Well, if we take that spiking activity from the brain and we just try to directly decode it, let's try to decode the movement that somebody's trying to make. Here's how we do. So here's an example of a few different conditions where the monkey was reaching. So here, the monkey's reaching down into the right, and you see each of these lines is a different reach. So there's a little bit of variability from reach to reach. Here up and to the left, here down and to the right in a really curved fashion. And if you just try to decode this activity, so now I'm just showing you kind of the single trial decoding from the spiking activity, you can see that it sort of gets the direction right. So it's these trajectories are pointed in the right direction, but the details of the movement aren't great. So kind of our signal to noise here is not very good. And we can quantify that by asking how much of the velocity of the hand can we reconstruct, and we're at about 70%. So decent, but not great. But if instead we first model those dynamics and then try to decode those denoised representations that the autoencoder pulled out, we can do quite a bit better. So capturing about 90% of the variance of the hand velocity, and you can see the trajectories pulled out from decoding are a lot smoother. They seem to be a much closer match to the actual trajectories that the monkey made. Okay, so hopefully that shows you that here's an application of machine learning and AI where we can better understand movement intention from neural activity. What I want to show you is kind of results along the lines of a different problem, which is that, and maybe this is a plea to the sort of materials experts in the room. One of the biggest challenges with neural interfaces is that our recordings are not stable, right? So, for example, we've got this Utah array that's a silicon microelectrode, like manufactured using standard like silicon manufacturing techniques, really stiff. We drop that into the brain, which kind of has the consistency of jelly, and it's constantly moving around due to respiration and blood flow. And you have this constant movement between this jelly-like substance and a really stiff object. It means we don't have a stable interface here. So we might be recording from some number of neurons on a given day. We come back, maybe a day later, maybe even an hour later. And we might not see that neuron anymore because the electrodes have changed positions or some scar tissue has developed or something like that. And we might, on other days, see a different neuron has kind of come into the window. So this becomes a really challenging decoding problem. Think about like if you were doing image classification, and I kept scrambling the pixels on you, right? I kept changing the input to the system. That becomes really hard. Like your classifier is not going to work very well when we scramble the pixels, and that's what we see, with neural interfaces. So let's say I learn a decoder from some recording, and this is, the monkey in this case made 8 directions of movements. I've colored them by direction, and then we come back the next day, and all of a sudden the recording configuration has changed. Let's try to use that same decoder, and you can see that it basically collapses because its input has changed. So it's not doing a good job of reconstructing behavior anymore. So what can we do to try to address this problem? Well, the kind of standard approach is like, let's say a person's using the brain-computer interface, and maybe the input signal has drifted a little bit, so it stops working very well. We just have them stop. Stop using it. We're going to take a bunch of new training data and recalibrate the system. And you wouldn't want to use any system where you're typing on your keyboard and all of a sudden it's spitting out garbage, and you have to relearn the mapping from fingers to keys, right? Like, we want it to just work. So how can we deal with that? Well, I showed you that neural populations exhibit these underlying dynamics, right? This idea that there are these kind of lower dimensional manifolds, there are these rules. The really nice property about that is that that's a property of the entire system, of the entire network, not the specific neurons that we're recording from, right? So I might be recording from these 3 neurons on one day, and I find these dynamics. Those dynamics correspond to any other neurons that I'm recording from in the same area. So if the electrodes shift around, I need to just figure out how to map down to that same state space, and I should be able to read out behavior with the same accuracy I had before, and the same for any other set of neurons that I'm recording from a given area. So how do we solve that problem? Well, the key point here is, even though the input is changing, there's a representation here of the network's activity that is stable. It has a stable relationship with the intended movement. So if we can find that and keep kind of remapping to that, we can keep these BCIs stable. Okay, so how do we do that? So we know, on the first day, our training day, we can find some underlying neural manifold. Then for any kind of future data where maybe the input has changed, the neurons we're recording from are different, we'll get some, output that does not look, we'll get some low-dimensional representation that does not look like what we had before. But we can use unsupervised alignment strategies to just say the output of the system should have the same distribution of activity that it had before. We don't need to know exactly what the behavior was doing. We just know that, like, the distribution of latent activity was here, and now it's over here. So the system just needs to pull it back, okay? And we do that by really just, we have a model, or a current neural network model of the system's dynamics. We just freeze that because we think those dynamics are the same all the time. And all we're trying to do is think about like rotating the input in order to get the system to get back to the original trajectory. So the trajectory was messed up. The objective of the system is to now find a rotation of the input through a feed-forward network that allows the distribution of these latent trajectories to match. Okay, so practically what does it look like? So if we have these latent trajectories on one day, we come back on a later day and you see that they've collapsed. They don't have the same structure anymore. And what that means is if this is the actual behavior that the monkey was performing, but you tried to decode using the same decoder, it would not work very well. So in terms of quantifying accuracy, this R-squared is just very, very bad. Okay, but if we do this kind of unsupervised alignment process using what we call nonlinear manifold alignment via dynamical systems, here I'll just show you like what happens during the training process. So the loss function here is effectively telling the system that its output needs to match the original latent distribution. And you can see that through the training process, that's what happens. So these kind of collapsed trajectories end up being pushed out to match the original trajectories. But the key thing here is this is all unsupervised. So the system doesn't know anything about what behavior is actually being performed here. It just knows that the distributions don't match. Once it makes them match, we can then decode this activity with the same original decoder that we trained on the original training set, and it does much, much better than it was doing before. So we've shown that this can work over very long timescales. So for example, let's imagine that you had a BCI and you didn't recalibrate it for 3 months and then came back. You could use this approach to rescue performance. So here, this is y-axis here is R-squared across many days, like how long has it been since you trained your decoder. If you just kept things static, not very good. But if you use this recalibration approach, you can recalibrate even out to 3 months and likely longer, because there's a lot of neuroscientific literature that says that these manifolds can be stable over years-long timescales. Okay, so hopefully I've convinced you that there's some nice applications of AI, and especially when we combine AI with insights from the systems neuroscience literature, we can achieve higher performing brain-computer interfaces and BCIs with improved robustness. So I definitely want to thank funders and the fantastic folks in the lab that made this happen. Thank you very much.
Thanks. We have time for a few questions. Yes, sir.
Yep, I was wondering,
Hold on. She's coming.
Thank you very much. So I have a quick question. I was wondering about if you can learn what you train on one person and then use it on another person.
That's a fantastic question. So, and there's so much in that question. It's kind of, how similar are the data between people? So let's start kind of at the highest level there. Like, let's say I implanted electrodes into one person and implanted electrodes into another person. Well, we know kind of at a basic level, like the neurons we're recording from are not the same. So it's very unlikely that you could just use the exact same decoder, without modification from one to the next. There are a couple ways, though, of leveraging information, I think, to do this in like kind of transfer learning approaches. So one is that these structures that I showed you, these low-dimensional structures, these dynamics, are likely to be conserved, and we do see similarities across people. So that does give us kind of a substrate that could be similar. I think we're going to need, some larger data sets than we typically have in neuroscience to be able to kind of test out those hypotheses. But yeah, I think there's a lot of power there. There are some great approaches that have recently been developed. So based on transformers, so like attention mechanisms can be permutation invariant or permutation equivariant, which means if you kind of change the input, the attention mechanism can still give you consistent output. So that's really, really useful for the problem I was just talking about. It's challenging because those models get really big. As you know, like transformers can get to be pretty big, but there's definitely, I think, some great work there from Eva Dyer's lab and from some folks at Pitt. And then the last thing I'll say is that we also know that if you pre-train on one subject, you can at least lower the data requirements for your next subject. So if you fine-tune on data from another subject, like you can learn a lot of structure that's useful in the transfer learning problem. Great question, thanks.
There?
So, hi, Professor. I have two questions. So, the first one is very fundamental. So, how did you distinguish 200 neurons from a U-turn? Were you using a threshold or some other method, which I don't really know? And second one is, do you see a future if I go single neural activity or single neural resolution using some other electrode for brain-machine interfaces, like a larger coverage instead of a very local?
Sure. Yeah, great questions. So the first question, so I showed you the example with 200 neurons, and those were actually like 200 sorted units, meaning some poor grad student had to sit there and very carefully make sure that they're extracting a given neuron, that it's stable over time. Not something that you want to do for brain-computer interfaces. One thing that a few folks have noticed is that we don't need that single neuron resolution. Like we don't need to track neuron identity, but even if we're like not sorting but just using threshold crossings, for BCI applications it actually works fairly well. So that's a really information-rich signal, even if you don't know like neuron A versus B versus C and just kind of lump them all together. I think the other question was about different types of interfaces, and maybe there's sort of two directions you could go with that. So one, I think there's a lot of impressive work in the electrocorticography space where you have larger contacts and they're typically on the surface of the brain, so rather than being implanted, or stereo electroencephalography also has larger contacts, but that's within the brain surface itself. It is I'd say, I don't know that they'll get to the same kind of level of resolution, like information richness that we can get from intracortical electrodes, but we do have really powerful AI methods, so maybe you don't need it. I think that's an open question. The other thing is, I think there's a lot of fantastic interfaces being developed right now. I mean, here at Rice, you guys have Lan and Chong developing flexible interfaces. I'm really excited by the stuff that Neuralink has been doing. Paradromics has been doing some great work. There's a ton. So I could see it going either way. I think for these types of applications where you really want high performance, you want real time, I think high resolution interfaces, especially if they're flexible, could really do a lot in this space.
Question there?
Thanks for the talk. That was really interesting. So I wanted to ask, I guess this is kind of a technical question. I don't know if it's applicable. Sure. So like the process you described is deterministic, and I know that for like sometimes like for hand motions and certain motions, you can break them down into like 5 or 6, main gestures. But I was wondering if it's actually more, have you considered using a more stochastic model, like, variational autoencoder? Because I know you're using an autoencoder, but they're a little more, your process is deterministic, but I don't know if like a stochastic model might be better for these kind of situations, especially if you get to like speech, where I don't know if it's more stochastic process, I guess.
It's a really good question. I think there are a lot of different levels of that question, but it's really interesting. So one, I glossed over it. This was a variational autoencoder. Okay. But that's really, for training purposes, like it trains pretty well using, variational methods. The dynamics model, so the recurrent neural network that takes in the vector and produces the trajectory, that was deterministic. So there's no kind of noise there. And we find, kind of, even though you learn a distribution over your latents, even if you just take the mean after training, it does about the same thing as like sampling from that distribution many times and averaging. I think maybe a really interesting part of your question is like, what if you made that dynamics model stochastic? Yeah. Would that allow you to, like we're treating a system that has a lot of noise in it as a deterministic system? I really don't know. People have different opinions on this. I don't know how much noise there is in the brain, especially like when you're doing motor control and like you don't actually want a noisy system. Like you're trying to very rigid. It is a controls problem. So I don't know. I mean, like I think the jury's still out. I think stochastic models have a lot of application in things like decision making and things like that. But I haven't seen really convincing evidence of kind of noise in motor dynamics. But really great question.
Sure. Thank you.
One last short question. Ananya?
Actually, I have a two-part question.
Oh, God.
Yeah. So, I was very curious to know how these low-dimensional manifolds vary with the progression of a neurodegenerative disease. And in case if you apply any form of rehabilitation therapy, any kind, how do they progress? Do they come back to what they were before the onset of the neurodegenerative disease, or do they take some other trajectory?
I think that is a fantastic question. I would really love
my
it.
student.
Oh, okay. All right, that explains it. I would really love it if somebody answered that. I certainly don't have the answer to that, but I think I like to think that these low-dimensional manifolds and dynamics give us something that we could look at in general, like over long timescales. Like when we're recording individual neurons, and especially with these interfaces where you don't have the same neuron over time, it's hard to know, like if I come back 2 weeks later, is the change I see due to the, we're recording different neurons, or is it due to the system has changed because of learning or because of degeneration or something? So I do think these like low-dimensional representations give us something to compare across days, but I don't know that anybody's, I don't know that literature well enough to know like kind of where the jury is on that one, but great question.
Thank you so much.
Thank you.
Thank you.
Chethan.